வாகை சூடவா.
Dear students, this class is a continuation of Mendelian experiments. According to Mendel, a gene or a factor or pair of genes controls a phenotype. More than a pair of genes or more than a pair of alleles controls the phenotype or physical expression of a character, then Mendelian ratios gets modified. In this class, we are going to study about intragenic interaction and intergenic interaction. You know, gene interaction, intragenic, before going that, you should have a clear idea about the locus. This is chromosome with centromere and with the chromatid. This is centromere, this is chromatid. And the position of a gene or an allele in a chromosome is said to be locus. The position of a gene or an allele in the chromosome is said to be locus. Intragenic interaction means the interaction between the genes present in the same locus. Intergenic interaction means the interaction between the genes present in two different loci. This kind of interaction responsible for modification of Mendelian ratios 3:1, 9:3, 3:1 or all gets modified into incomplete dominance. For example, in intragenic interaction, incomplete dominance is a very good example. The ratio incomplete dominance in Mirabilis jalapa and snapdragon, the ratio is 1:2:1. Codominance, ABO blood group, where the interaction is 1:2:1. Like that, in intergenic interaction, the interaction between the genes present in two different loci. Singular is locus, the plural is loci. So the genic interaction takes place between two genes present in two different loci. Different ratios, different modifications are seen. These modifications, now known as genic interaction or factor interaction, where Mendelian ratio, dihybrid cross ratio 9:3:3:1 gets modified into either 9:7 or 12:3:1 or 13:3 or 15:1, like that. What is gene interaction? Either inter or intragenic interaction. More than a pair of genes or more than a pair of alleles controls the phenotype, then Mendelian ratio 9:3:1 gets modified. We are calling it as genic interaction or factor interaction. Some of the important ratios needed for your NEET examination regarding intergenic interaction. Number 1, dominant epistasis, the ratio is 12:3:1, reported in cucurbita pepo, the summer squash plant. Second is recessive epistasis, reported in Antirrhinum majus plant. The common name is snapdragon, 9:3:4. Duplicate recessive or duplicate genes, the recessive duplicate genes, the ratio is 9:6:1. It is also reported in the fruit color of cucurbita pepo, the summer squash plant. Then complementary gene in Lathyrus odoratus, sweet pea. You know, the common name of Lathyrus odoratus is sweet pea plant. The ratio 9:3:1 gets modified into 9:7. The next one is supplementary gene reported in maize. The ratio is 9:3:4. The next one is duplicate gene. Here 9:3:1 gets modified into 15:1. All these ratios are important for you in the NEET examination. Then blood group. Human blood group is a very good example for codominance. This blood group first reported by K. Landsteiner, the scientist K. Landsteiner. The genes controlling the blood group are capital A, capital B and O. Scientifically, we are calling it as IA gene, IB gene and IO genes, they control the blood group. The genotype of blood group A is they have either IA, IA or they have IA, IO. You know, A and B are the dominant genes and O is the recessive gene. And the blood group, the genotype of blood group B is they have IB, IB or IBIO. Here I stands for isohemagglutinin and the blood group O is IOIO. But in the blood group AB, A and the B genes are present and their characters expressed. This phenomena is said to be codominance. See the chart, you know the phenotype and genotype. Phenotype of blood group A is A, B, O and AB. The genotype is IAIA or IAIO, IBIB or IBIO, and the third is IOIO, the fourth is IAIB. The AB blood group is said to be the universal recipient and the O blood group is the universal donor. These two things important for your NEET examination. So, universal donor in the blood group is O, the universal recipient is AB. And in A and B blood group, they have antigen A and antigen B. In O blood group, antigens are totally absent. AB blood group with A antigen as well as B antigen. Antigens are totally absent in O blood group, like that antibodies are totally absent in AB blood group. These things you should remember for your NEET examination and they ask questions in this topic. Then RH factor, also known as erythroblastosis fetalis or hemolytic disease. This disease reported by Lance Steiner and A.S. Wiener and scientifically described by Race and Fisher and Macaca rhesus, the monkey Macaca rhesus. The term RH derived from the monkey Macaca rhesus. This gene, capital D gene, responsible for RH factor causes a disease in human system called erythroblastosis fetalis or hemolytic disease. If RH positive male marries a RH negative female, the fetus will be always RH positive in nature because it receives capital D gene from father and small d gene from the mother. The fetus is always positive in nature, but the mother is negative in nature. So, the D gene or D antigen present in the bloodstream of the fetus enters the bloodstream of the mother. To kill the foreign antigen, to eliminate the D antigen, mother's blood produces antibodies. These antibodies are not sufficient in strength to kill the first fetus. If the mother subsequently carries RH positive fetuses, then the fetuses are at risk by a disease called erythroblastosis fetalis or hemolytic disease. It is an important one for our NEET examination. Then positive male marries negative female, then the fetus will be positive in nature, then it at risk by a disease called erythroblastosis fetalis or hemolytic disease. They to ask this kind of questions in our NEET examination. Then is sex determination. Different types of sex determination reported in animals. One is XX XO type of sex determination. Sex determination, the sex chromosomes, we have two different types of chromosome. One is the autosome, another is the sex chromosomes, also known as the allosomes. The genes present in the autosomes controls the body characters. The genes present in the sex chromosomes controls the reproductive characters. XX, XY, like that, these are all the sex chromosomes which control the sex. XX, XO type of sex determination reported in bugs, insects like cockroach and grasshoppers. Here, XX, similar sex chromosomes are present in female and XO, different types of sex chromosomes are present in male. So, XX, XY type, XO type of sex determination reported in bucks. Then second is XX, XY type of sex determination. XX, XY type of sex determination reported in man. Actually, in female, 44 plus XX similar sex chromosomes are present. In male, 44 plus XY different types of or dissimilar sex chromosomes are present. At the time of gamete formation, in females, similar gametes are formed, namely 22A plus X or 22 plus X and 22 plus X. So, females said to be the homogametic individuals, but in males at the time of gamete formation, two types of gametes are formed, namely 22 plus X and 22 plus Y. So, males are known as heterogametic individuals. This kind of XX, XY kind of sex determination reported in man as well as Drosophila melanogaster, the fruit fly. The next one is heterogametic females. Sometimes here in these cases, females are homogametic in nature, males are heterogametic in nature. But certain animals, the females are heterogametic in nature and males are homogametic in nature. See here, heterogametic females are seen in insects. Certain insects, they have heterogametic females. See the chart, ZO, Z is a type. This method of sex determination seen in certain moth, butterflies and chickens. Here the female AAZO, see here, AA represent autosomes, ZO represent the sex chromosomes. They produce two different types of gametes, so X and O. So, females are heterogametic in nature. Here males are Z. See here, Z, they produce similar gametes. This is a homogametic male. Next is ZW. Z is a type of sex determination reported in gypsy moth, certain fishes, reptiles and the birds. And sex determination already I explained to you. The next one is haplodiploidy in honeybees. What is a haploid? What is a diploid and what is a polyploid? You know these things. Somatic cell of an organism with only one set of chromosome is said to be haploid. Somatic cell of an organism with two sets of chromosome is said to be diploid. The somatic cell of an organism with more than two sets of chromosomes, the condition is said to be polyploid. In haplodiploidy condition reported in honeybees. In honeybee, the female undergo oogenesis. The development of egg or ovum in the ovary is said to be oogenesis. In the end of oogenesis, the egg is formed. In the male, at the time of spermatogenesis, the male gametes are formed. The male and the female gamete undergo sexual fusion or fertilization, results in the formation of zygote. The fertilized egg in honeybee develops into either worker bee or the queen bee. The unfertilized egg develops into male. See here, fertilized egg, the zygote, develops into embryo. It will become either worker bee or the queen bee. The unfertilized egg, this is said to be parthenogenesis. Development of a new organism without the act of fertilization is said to be parthenogenesis. So, parthenogenesis reported in honeybee, and males of honeybee is always haploid in nature. This phenomena is said to be haplodiploidy in honeybees. This is also important for your NEET examination. Next one is sex-linked inheritance. This is very important. Sex-linked inheritance means we have two types of chromosomes, you know pretty well boys, pretty well students. One is two different types of chromosome. One is autosome, which control the body character. Remember, autosomes always controls our body character. Like that, another one is allosome. The sex chromosomes controls the reproductive characters. In some cases, the genes present in the sex chromosomes are allosomes. The genes present in the sex chromosomes or the allosomes control the body characters. Then they are known as sex-linked genes, and their inheritance is said to be sex-linked inheritance. The genes in the autosomes controls body characters. The genes present in the sex chromosome controls the reproductive characters. If the genes present in the sex chromosomes controls the body character, then they are known as sex-linked genes, and their inheritance is said to be sex-linked inheritance. Two types of sex-linked inheritance reported in man. One is X-linked inheritance, another is Y-linked inheritance. If the genes present in the sex chromosome or X chromosome control the body character, then they are known as X-linked genes, and their inheritance is said to be X-linked inheritance. Color blindness is a very good example for this one. So, if the genes present in the X chromosome control the body character, then they are known as X-linked genes, and their inheritance is said to be X-linked inheritance. If the genes present in the Y chromosome control the body character, then they are known as Y-linked genes, and their inheritance is said to be Y-linked inheritance, and Y-linked genes are also known as holondric genes. Certain examples given for the sex-linked inheritance in our book. Next is inheritance, sex-linked inheritance. The best one is hemophilia or Bledo's disease. What is hemophilia or Bledo's disease? Normally, the blood clots within 2 to 8 minutes. If it takes 30 minutes to 24 hours, failure in blood clotting mechanism is said to be hemophilia or Bledo's disease. It's a very good example for X-linked inheritance. The next one is color blindness or achromatopsia. What is night blindness? What is color blindness? If you are unable to differentiate colors in the dim light, we know night blindness, achromatopsia, color blindness, night blindness is said to be, we have two diseases. One is color blindness. Color blindness is said to be achromatopsia. Another is night blindness. Night blindness technically known as nyctalopia. We can treat this night blindness because it is due to vitamin A deficiency. But color blindness or achromatopsia means you are unable to differentiate certain colors in dim light. It's a very good example for X-linked inherited character. This color blindness is of two types. One is protonopia, another is deuteranopia. If you are unable to differentiate red color in dim light, we are calling it as protonopia. Green color in dim light is said to be deuteranopia. These two important for your NEET examination. It's a very good example for X-linked inherited characters. We can treat this color blindness only through gene therapy, but we can treat this night blindness or nyctalopia with the help of vitamin A substitution. Then next is inheritance of Y-linked character, holondric genes. The best one is hypertrichosis, excessive development of hairs and pin of the hair. It's a very good example for Y-linked inheritance. Y-linked genes, the holondric genes, it is common in certain families. Next is karyotyping. The diagrammatic representation of chromosome is said to be karyogram, and the method helps in arranging the chromosome is said to be karyotyping. Actually, 7 different groups of chromosomes are present. So, karyogram, diagrammatic representation of chromosome is said to be karyogram or ideogram. The methodology helps to arrange the chromosome in a particular manner is said to be karyotyping. We have 7 different groups of chromosomes: A, B, C, D, E, F and G. These things are important for your NEET. And the different types of chromosome, actually 4 different types of chromosomes reported in man. One is telocentric, another is acrocentric, third is submetacentric, and fourth is metacentric. These are important for your NEET examination. Then next one is Mendelian disorders. All the things against law of dominance or law of segregation or law of independent assortment is said to be Mendelian disorder. Mendelian disorder mostly due to mutation. What is mutation? Sudden spontaneous deviation from the wild type is said to be mutation. In human, various mutated diseases are reported. The diseases are known as congenital diseases. The diseases inherited from parents to the subsequent generations are known as the heritable diseases, also known as the congenital diseases. Sickle cell anemia, thalassemia, agammaglobulinemia, albinism, Huntington's chorea, severe combined immunodeficiency disease. These are all example for Mendelian disorders. These disorders mostly due to dominant or recessive gene mutation. Sometimes gene or point mutation is also responsible for Mendelian disorders or heritable diseases. This gene or point mutation is very important to learn the higher order genetics. What is gene or point mutation? Sudden change takes place in a nucleotide or a pair is said to be gene or point mutation. You know, nucleotide, the nanoparticle of living organism. Nucleotide chemically composed of nitrogenous base, sugar and phosphate. Adenine always pass with thiamine, guanine always pass with cytosine. First the nitrogenous bases combines with sugar to form a nucleoside. Nucleoside combines with the phosphate molecule to form a nucleotide. Many nucleotides unite to form polynucleotide. Finally the nucleic acid is formed. Sudden change takes place in a nucleotide or a pair. We are calling it as gene or point mutation. So, three types of gene or point mutation reported in organisms, namely number 1 deletion, number 2 addition, and number 3 substitution. Removal of a nucleotide or a pair is said to be deletion. Addition of a nucleotide or a pair is said to be addition. One is substituted by the other is said to be substitution. Removal of a nucleotide, deletion. Addition of a nucleotide, addition. One nitrogenous base is substituted by the other, we are calling it a substitution mutation. All these mutations responsible for heritable diseases. This substitution mutation is of two types, namely number 1 transition and number 2 transversion. Transition means a purine base is replaced by a purine or a pyrimidine base is replaced by a pyrimidine. We are calling it as a transition mutation. Adenine is replaced by guanine or thiamine is replaced by cytosine. We are calling it as transition mutation. A purine base is replaced by a pyrimidine base or a pyrimidine base is replaced by a purine base. We are calling it as transversion mutation. A purine is replaced by a pyrimidine or a pyrimidine is replaced by a purine. We are calling it as transversion mutation. This substitution mutation is genetically important because it causes phenotypic changes in organisms. All these mutations causes heritable diseases in man. Then very good example for this one is albinism. Albinism, you know, complete lack of melanin pigment in the skin is said to be albinism. It's a heritable disease, you know pretty well. Like that Huntington's chorea. Huntington's chorea, first dominant gene mutation reported in human, like that aneuploidy. Autosomal aneuploidy as well as sex chromosomal abnormality causes syndromes in human. What is aneuploidy? Variation that involves chromosomal number within a set is said to be aneuploidy. Variation that involves chromosomal number within a set is said to be aneuploidy. This aneuploidy is of two types, namely one is a hyperploidy, another is hypoploidy. Hyper means increase, hypo means decrease. Addition of one chromosome 2n plus 1 trisomy, addition of 2 chromosomes tetrasomy, removal of 1 chromosome monosomy, removal of 2 chromosomes nullisomy. These are called aneuploidy. So, variation that involves chromosomal number within a set is said to be aneuploidy. It is of two types. One is hyper, another is hypo. Hyper means increase, hypo means decrease. 2n plus trisomy, 2n plus 1 trisomy, 2n plus 2 tetrasomy, 2n minus 1 monosomy, 2n minus 2 nullisomy. These autosomal as well as sex chromosomal ploidy, aneuploidy causes number 1 the Down syndrome, number 2 the Klinefelter syndrome, and number 3 the Turner syndrome. Down syndrome is a very good example for autosomal aneuploidy. The 21st pair have 3 chromosomes instead of 2. This is called autosomal. And this Klinefelter syndrome and Turner syndrome example for the formation of transgenders. Irregular meiosis or non-disjunction of sex chromosomes, irregular meiosis or non-disjunction of sex chromosomes responsible for this Klinefelter syndrome or Turner syndrome. See here, a male with 44 plus XY chromosome, female with 44 plus XX chromosome. In male, if irregular meiosis takes place, you get the gamete like this, 22 plus XY and 22 plus 0. In female, regular meiosis takes place, 22 plus X and 22 plus X. See the gametes in males. Here, the autosomes alone undergo meiotic cell division. The sex chromosomes are not separated. This non-disjunction or irregular meiosis responsible for the formation of one male gamete with additional chromosome, another male gamete with less chromosome. One gamete with 24 chromosome, another male gamete with 22 chromosomes. This is called irregular meiosis or non-disjunction of sex chromosomes. This male gamete fuses with this female gamete results in the formation of 44 plus XXY, Klinefelter syndrome, male sterility, and this male gamete fuses with this female gamete produces 44 plus XO, Turner syndrome, sterile females. So, this Klinefelter syndrome, Turner syndrome, Down syndrome or 21 trisomy, thalassemia, sickle cell anemia, A gamma globulinemia, everything important for your NEET examination. Thank you students, I hope this class is very useful for you. Thank you. Dear students, welcome to the next genetics class. In genetics, the remaining parts in genetics, the ploidy, the polyploidy, autopolyploidy, allopolyploidy, mutation, replication of DNA, and the genetic code. These topics are important in advanced genetics. In this class, I like to discuss some of the important questions for your NEET examination. Question number 14. In a hexaploidy wheat, the haploid N and basic X numbers of chromosomes are. In a hexaploidy wheat, wheat, Triticum vulgare, the haploid N and the basic X numbers of chromosomes are option A, N is equal to 21 and X is equal to 7, option B, N is equal to 7 and X is equal to 21, option C, N is equal to 21 and X is equal to 21, and option D, n is equal to 21 and x is equal to 14. This hexaploidy wheat triticale is the first man-made cereal. Hexaploid wheat, the triticale, first man-made cereal. It is an example for allopolyploidy. You know pretty well, polyploidy means the somatic cell of an organism with more than 2 sets of chromosomes. If the somatic cell of an organism with more than 2 sets of chromosomes, we are calling it as polyploidy. This polyploidy is of 2 types, namely one the autopolyploidy, another one is the allopolyploidy. Chromosomal set number increased in a hybrid produced by crossing 2 different plants. This triticale, developed by the scientists in the lab, it is the first man-made cereal. This triticale developed by crossing Triticum durum and Cigale cereal. Triticum durum with 28 chromosomes crossed with Cigale cereal with 14 chromosomes. At the time of gamete formation, Triticum durum donates 14 chromosomes and Cigale cereal donates 7 chromosomes. This Triticum durum and Cigale cereal, male and the female gametes are fused. We get a new plant called triticale with 21 chromosomes and it is sterile in nature. The scientist doubled the chromosome number triticale by using the chemical colchicine. The main function of colchicine obtained from Colchicum autumnale plant is to double the chromosome number. Finally, we get the hexaploid triticale with 42 chromosomes. So, your question is 28, 7, 21, 14 plus 7, 21, 42, it is a plant, fertile triticale plant with 42 chromosomes. Now the question is, in a hexaploid wheat, here this is hexaploid wheat, the haploid n and the basic number of chromosomes are, here see this Cigale cereal, it donates only 7 chromosomes, it represents X. Here this 21 triticale, it is doubled using colchicine, this represents N, this represents 2N. So the correct answer for the question number 14 is option C, N is equal to 21 and X is equal to 7. So this triticale, allopolyploidy, is a very good example for allopolyploidy. Allopolyploidy means chromosomal set number increased in a hybrid produced by crossing 2 different species. One is Triticum durum, another is Cigale cereal. So the answer is option A, N is equal to 21 and X is equal to 7. Question number 15, point mutation involves. Already I informed about this gene or point mutation. Sudden change takes place in the nucleotide or a pair, we are calling it as gene or point mutation. It is of 3 types, namely deletion, addition and substitution. And deletion, removal of a nucleotide, addition of a nucleotide, one nitrogenous base is substituted by the other, we are calling it a substitution mutation. Change in base pair, we are calling it as frame shift mutation. Duplication is nothing but the small portion of the chromosome is doubled. So in this question, point mutation involves option A, deletion, option B, insertion, option C, change in single base pair, and option D, duplication. The correct answer for the question number 15 is option C, change in single base pair. Single base pair means sudden change. See here the question number 15. Sudden change takes place in a nucleotide or a pair, we are calling it as gene or point mutation. The perfect answer, see the question 15 once again, point mutation involves deletion, insertion, change in single base pair, duplication. In this option A, Deletion represent the deficiency, deletion or deficiency. If a small portion of the chromosome, it's an example for chromosomal aberration or chromosomal abnormality or chromosomal mutation. If a small portion of the chromosome is removed, we are calling it as deletion or deficiency. Option B, insertion. If a small portion of the chromosomal segment is added, we are calling it as insertion. Duplication means if a small portion of the chromosome represented twice, see here A, B, C, D, see here A, B, C, D, C, D. If a small portion of the chromosome represented twice, we are calling it as duplication. Option C, change in single base pair. This represent substitution. This represent substitution gene or point mutation. One nitrogenous base is replaced by the other, we are calling it as substitution mutation. Purine by a purine or a pyrimidine by a pyrimidine is called a transition. Purine by a pyrimidine or pyrimidine by a purine is said to be transversion. So the correct answer, perfect answer for the question number 15 is option C, change in single base pair. This represent substitution. It may be either transition or transversion. Transition, purine by a purine, pyrimidine by a pyrimidine, or purine by a pyrimidine or pyrimidine by a purine, we are calling it as transversion. This is the opt answer for the question number 15, point mutation. Then question number 16, which one of the following is a wrong statement? See the question once again, which one of the following is a wrong statement regarding mutations? Mutation, you know, sudden spontaneous deviation from the wild type. Somatic mutation, gametic mutation, forward mutation, backward mutation, biochemical mutation, lethal mutation, progressive mutation, retrogressive mutation, gene or point mutation, chromosomal aberration, like this various kind of mutation reported in plants as well as animals. Question is, which one of the following is wrong statement regarding mutation? Option A, UV and gamma rays are mutagens. Option B, change in a single base pair of DNA does not cause mutation. Option C, deletion and insertion of base pairs cause frame shift mutation. Option D, cancer cells commonly show chromosomal aberrations. Question is, which one of the following is a wrong statement regarding mutation? Option A, UV and gamma rays are mutagens. Yes, mutagens means the substances or the chemicals or the rays used to stimulate, used to induce mutation. So mutagens means mutation inducing substances. Ultraviolet rays as well as gamma rays causes mutation in plants as well as animals. Change in a single base pair of DNA does not cause mutation. No. Change in a base pair, single base pair is also causes mutation. Option C, deletion and insertion of base pairs cause frame shift mutation. See, option D, cancer cells commonly show chromosomal aberration. The correct answer for the question number 16 is option B, change in single base pair does not cause mutation. No, change in single base pair, adenine by guanine or thymine by cytosine or adenine by thymine or cytosine by guanine causes mutation. But here the answer is does not cause. So it is the incorrect statement, it is the wrong statement. So the correct answer for the question number 16 is option B, change in single base pair of DNA cause mutation, not does not cause mutation. So this statement is incorrect. Question number 17, which of the following statements is not true of two genes that show 50% recombination frequency? Which of the following statement is not true? See, the question is not true of two genes that show 50% recombination frequency. You know, recombination is the end product of crossing over. Law of independent assortment, according to Mendel, the parental combination should be 50%. The recombination should be 50%. The ratio is 1:1:1:1 dihybrid cross. This recombination frequency may vary. In coupling experiment or a repulsion experiment, in coupling experiment the parental combination is more. The repulsion experiment the recombinants are more. See the question, which of the following statement is not true? Option A, the genes may be on different chromosomes. Option B, the genes are tightly linked. Option C, the genes show independent assortment. Option D, if the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. The correct answer for the question number 17 is option B, the genes are tightly linked. See the question, which of the following statement is not true of two genes that show 50% recombination frequency? The answer is the genes tightly linked is incorrect. Loosely linked is the correct one. Then question number 18. Haploids are more suitable for mutation studies than the diploids. This is because, you know, haploids, the somatic cell of an organism with one set of chromosome. Diploid, the somatic cell of an organism with two sets of chromosome. Now come to the question, haploids are more suitable for mutation studies than the diploids. This is because option A, all mutations whether dominant or recessive are expressed in haploids. Dominant means the character expressed in the F1 generation. Recessive means the character which is not expressed or suppressed in the F1 generation. Option B, haploids are reproductively more stable than diploids. Haploid as well as diploids are stable. And option 3, mutagens penetrate in haploids more effectively than diploids. Mutagen, you know, mutation causing substances. Option D, haploids are more abundant in nature than diploids. The correct answer for the question number 18 is option A, all mutations whether dominant or recessive expressed in haploid. See the question, haploids are more suitable for mutation studies than the diploid. This is because all mutation, it may be dominant or recessive, very easily expressed in haploids. It also represents the gametes. Then question number 19, crossing over that results in genetic recombination in higher organisms occurs between crossing over the results of genetic recombination in higher organisms occurs between option A, non-sister chromatids of a bivalent, option B, two daughter nuclei, option C, two different bivalents, option D, sister chromatids of bivalent. You know crossing over. Actually crossing over is a process takes place during the Packettine stage of prophase 1 of meiosis 1. Meiosis, the reduction cell division. During meiosis 1, prophase 1, the Packettine stage, the important phenomena crossing over takes place. Before going to the answer, you know some clear idea regarding this concept. During zygotine stage, pairing of homologous chromosomes takes place. Pairing of homologous chromosome is said to be synapses, and the paired homologous chromosomes are known as bivalents. During the pairing, one chromosome from the paternal side and other chromosome from the maternal side. This pairing of chromosomes takes place during zygonema, is said to be synapses, and the paired homologous chromosomes are called the bivalents. In the end of this zygotine stage or zygonema, it will become a tetrad. You know, tetrad centromere with 4 chromatids or chromosome with 4 chromatids is said to be tetrad. This is because of the replication of DNA. One chromatid replicate to form the another. It represents the replication of DNA. Replication is nothing but production of identical copies of the DNA. This is the original chromosome from the father, and this is the original chromosome from the mother. This is template. This exact replica, it is the end product of replication. This is original, this is replicated one. This side is original, this is replicated. The original chromatids come from the parental side, either from the paternal side or the maternal side, the chromatids present, the original chromatids are known as the sister chromatids. The newly formed chromatids, this one is sister original, this one is sister original. The newly formed chromatids are known as the non-sister chromatids. Crossing over actually takes place between the non-sister chromatids of homologous chromosome. The exchange of genetic material takes place between the non-sister chromatids of the homologous chromosome is said to be crossing over. So the correct answer for the question number 19 is option A, non-sister chromatids of a bivalent. Then question number 20. Removal of introns and joining the exons is defined order in a transcription unit is called tailing, transformation, capping and splicing. What is transcription? At the time of protein synthesis, you know protein synthesis, production of protein is said to be protein synthesis. Protein synthesis actually takes place in the ribosomes. So the ribosomes are known as the protein factories. Ribosomes, the protein factories, where the protein synthesis takes place. The amino acid united by, combined by peptide bond, dipeptide bond, polypeptide bond, finally a long chain of protein is formed or protein is synthesized. At the time of protein synthesis, the first process, transcription, takes place. Transcription is nothing but the conversion of DNA into mRNA. So production of mRNA, transcription is nothing but the conversion of DNA into mRNA or the formation of mRNA from the DNA. In the initial stages of mRNA formation, it is with the exons and introns. The coding region of DNA is said to be exon and the non-coding region is said to be intron. For example, this is a mRNA, newly formed mRNA. This area is said to be intron, intron, intron. This is intron, this is intron, this is intron. This coding region is said to be exon. Removal of intron, if you remove the non-coding region, finally you get only the exons. So removal of intron from the precursor mRNA is said to be splicing. So you remove introns and join the exons, the process is said to be splicing. So correct answer for the question number 20 is option D, splicing. So exon means coding region of mRNA or precursor mRNA. The non-coding region is said to be intron. Removing the intron and joining the exons is said to be splicing. Then next question, question number 21, selection. Select the correct option from the A, B, C, D options. See the question, direction of RNA synthesis. You know RNA is formed from the DNA. The conversion of DNA into mRNA is said to be transcription. Direction of RNA synthesis either from 5 prime or 3 prime, 3 prime 5 prime, 3 prime 5 prime, 5 prime 3 prime, you know the structure. If it is here 5 prime 3 prime, we are calling it as this side is opposite side is called 3 prime 5 prime. The correct answer for the question number 21 is formation of mRNA from DNA. Answer is 5 prime 3 prime and 3 prime 5 prime. See the question, 5 prime 3 prime and 3 prime 5 prime. The first option is correct, A, 5 prime 3 prime and 3 prime 5 prime. This is the correct answer. Before going to this concept, you should have a clear idea about DNA, the structure of DNA and the replication of DNA. The DNA is the genetic material and it carries hereditary information from one generation to the other. It controls protein synthesis. So function of DNA, it's a genetic material, carries hereditary information from one generation to the other. It controls all the metabolic activities of the cell and it also controls the protein synthesis. Peptide synthesis or protein synthesis inside a cell takes place in. Cell is a structural and functional unit of all living organisms. So inside the cell, the cell organelle involved in protein synthesis? Option A, ribosomes. Option B, chloroplastid. Option C, mitochondria. And option D, chromoplastid. The function of chromoplastid is to attract the insects for cross-pollination. The function of mitochondria is said to be the powerhouse of a cell where aerobic breakdown of food material takes place, aerobic respiration, the oxidation of carbohydrates, release of energy in the form of ATP, liberation of carbon dioxide and formation of water. So mitochondria is mitochondria where exothermic reaction, complete oxidation of carbohydrates takes place, energy is released and it is stored in the form of ATP. So ATP production actually takes place in the mitochondria. So mitochondria is said to be the powerhouse of the cell where conversion of chemical energy into kinetic energy takes place. Chloroplastid plays a vital role in photosynthesis. The main function of chloroplastid is to convert the solar energy into chemical energy. So function of chloroplastid, synthesis of starch. It is predominant in photosynthetic or green colored plants. And ribosomes, where the protein synthesis takes place. So correct answer for the question number 22 is ribosomes, the protein factories. And two types of ribosomes are present, already you know pretty well. One is 70S ribosome, another is 80S ribosomes. In plant and animal cell, 70S ribosomes present in the mitochondria and chloroplastid. 80S, the remaining cells have the 80S chromosomes. Like that, prokaryotic organism, primitive cellular organisms or organisms with an incipient nucleus is said to be prokaryotes. Example, bacteria and the cyanobacteria, blue-green algae, and the Archaebacterium, the bacterium which live in extreme environmental condition where 70S ribosomes are present. And 80S ribosomes are present in eukaryotes. U means true, karyon means nucleus. Organism with a well-defined nucleus is said to be eukaryotes where 80S ribosomes are present. So peptide synthesis, protein synthesis inside a cell takes place in. The correct answer for the question number 22 is option A, ribosomes, where protein synthesis, where peptide synthesis, where the union of amino acids takes place. So ribosomes are known as the protein factories. So the next question and the last question for this session is, during protein synthesis in an organism at one point, the process comes to halt. Select the group of 3 codons from the following from which any one of the 3 could bring about this halt. Halt represents the stop. You know codon. Codon means 3 nucleotides which serve as a code for an amino acid is said to be codon. So in protein synthesis, the role of codon is vital. 3 nucleotides which serve as a code for an amino acid is said to be codon. Here the question is stop or terminating codons, or the 3 could bring about the halt of protein synthesis. The correct answer for the question number 23 is option C, UAG. U represent uracil, A represent adenine, G represent guanine, UGA and UAA. I hope this question helps you to learn in applied genetics, advanced genetics. The remaining concepts and the remaining questions regarding genetics we'll see in the next class. Thank you.
இந்நிகழ்ச்சியை பற்றிய தங்களின் மேலான கருத்துக்களை கீழ்க்கண்ட முகவரிக்கு கடிதம் மூலமாகவோ மின்னஞ்சல் மூலமாகவோ குறுஞ்செய்தி மூலமாகவோ தெரியப்படுத்தலாம். அனுப்ப வேண்டிய முகவரி: சிறப்பு அலுவலர், கல்வி தொலைக்காட்சி, 8ஆம் தளம், அண்ணா நூற்றாண்டு நூலகம், காந்தி மண்டப சாலை, கோட்டூர்புரம், சென்னை 6085. தொலைபேசி எண் 7824015000.